Alright. Yeah. Okay. So let me start back again. I'll not take you much time. First of all, I know everybody is tired. We have seen a lot of things today. Okay. So I try to keep it minimum, maybe 30 minutes. Okay. So bear with me. So you have seen something right now that how the systems are working. What is the report that is coming? We'll be also demonstrating that tomorrow. पर इसके साथ में होता क्या है? आप एनजीएस बात करते हो, पढ़ते भी रहोगे आर्टिकल्स, राइट? पब्लिकेशंस देखते रहोगे या मोस्टली किसी से सुनते रहोगे. बट नॉर्मली एनजीएस में होता क्या है? एज एन एप्लीकेशन मैंने सुबह एग्जांपल दिया था कि ट्यून कर रहे हो, कोई कुछ चीज टारगेट कर रहे हो तो सीक्वेंस करना चाहते हो. तो जो वन वन बाय वन करना चाहते हो वो टुगेदर करना अच्छा रहेगा, राइट? साथ में करना बहुत इजी रहेगा. And that's how we like to work on that part. So how this could be useful? So normally once your data is generated as BAM, FASTQ, then he is more comfortable over there, I think. So, right, so rather, what we have is the QC, since this is giving me an opportunity to go around. So, you can do this. I can't give you. It's not working. Still, it's okay. So, what the opportunity is that once the data is generated, what can you do for it? How would you understand it? So, understanding you either need to do a QC, right? You need to understand how do you do a QC for Sanger? Any idea? How do you do a QC for your PCR reaction? How do you do that? Try to blast, try to see if it's similar or not. It gives you confidence and then you try to screen more. Right? So something like that is also available for the engines because engines will give millions of data, millions of reads coming at the same time. How could you screen that? So you have certain tools that are available, such as you have a FastQ tool. FastQ is a Windows-based tool which can be downloaded onto your system. Someday you outsource your data. Say, I'm giving an example. It gives you a data of BAM files or FastQ files. You can upload them into this tool on a Windows system. And it will start showing you how the reads are there, what are the qualities out of it. It will show box plots of the qualities. Because each position would have a quality value, right? So if I placed it onto x-axis of your sequence, okay, and see whether how the qualities are coming. So it would be good qualities and the bad qualities would drop at the end. So you know at what level the bad qualities would go, which length. So that gives you an idea whether your quality of sequencing is good or not. Okay. So with that, with this first part, it would be more of how the throughput would be helping you to know the basic uses. The ATGC content, the repeat sequences content. Okay. You need to know, is there any adapter remaining behind in your data? So it may happen the sequencing has happened, but the adapters are not cut. Is there any partial adapter remaining in your data? You would like to do that QC using fast QC. After that, if you want to remove certain adapters from the data, irrespective of the technology, if you feel some primers are remaining back into the sequences, what you have amplified, you want to trim that out. You can use a tool called as a FedAdapt. Okay, this one. Or else you have a FastX toolkit. So if you have FastQ files provided by your service provider or something that you are getting from system or you like to screen that more. So you see, this tool is called FastX toolkit. There are different commands to run inside. You can go through that one by one. But you see, when you have sequence, either you like to trim based on quality, either you like to screen them based on quality statistics. You need to do that because your data should be good quality so that you remove out errors from the data. Okay. So doing that using a fast text toolkit or else an NGS QC toolkit is also there. This is a command line tool. This is also a command line tool. Okay. So particularly, certain of these tools are embedded or put into the site that I told you in the morning. Use Galaxy. It's a free tool, but has all those modules being inserted into this part, this particular tool, use Galaxy. But of course, command line tools are also important. If you start studying bioinformatics, you like to use command line. Majorly. Good system with command line and these commands. Okay. With that, we'll start with the applications part. So first part that we are talking about is the 16S metagenomics. So metagenomics, it's more about the gene, 16S. It is spreaded and it has a certain variable region. You're studying the variable regions in such a way which is more specific towards each species. Okay? So lactobacillus would have its own variabilities in these ways. Target that and use that to know how well your sample is having microbial content. So targeting, you can see, tells me about a V2 region, which is the primer region. You have a V4 region, V3 region, again V6, 7, a longer one, around 280 to 90 base pairs is what you get. You have a V8 region, okay, it's a little smaller, and then the V9. So there are two sets. You can see the 1, 2, and 3 is one set of the primers. The second set of the primers is V3, 6, 7, and 9. Because you may have certain overlaps that may be coming. So these are two wires that are provided for the process. You must have processed two wires, merged them together, use that data again, and taken off equimolar to pull that together in one shape system. Okay? So you are doing that over here. So the overall idea is to target each and every variable region, get the data for each sample. With that data being taken up from the data, you are still looking further to merge that data with consensus. So your counts can be merged together. So your proportion could come further out of it. So you're doing a 400 base pair sequencing. Okay? Because you have a longer length region coming, 280 base pairs. So to be on the safer side. So two databases I told you. Green Genes database, you have microseeds database. You have multiple microorganisms present over here. Microseed is curated to the company itself. Whereas Green Genes is the one which has 4 lakh organisms into it. So it has all the variabilities that is present. So these would be used as databases. So whatever sequences you generate, cluster, get the proteus, that would be used over here to map and assign species, genus, and families to it. Okay. What would happen further? Something like this, a visualization. Okay, I'm not taking this, I'm taking further. Okay. There's something more that is coming further. I'll explain that in the next part. So you have something like the exome, which would actually help you to do exome analysis. So you have exome kit is there available. You can do a single sample analysis just to see what is happening different in those samples. It has the ion reporter software that has these workflows available so that you can analyze, amplify, sequence, analyze the ion reporter, you get to know what is different out of it. If you are doing something like pairing it, so pneumonormal or father mother child, you have a trial analysis that comes into play. Or you have looking at pneumonormal studies, paired exome studies, two sample together. Okay. So in this way, you have the workflows that are available that you can analyze for exome studies. So somebody who has lost a child would like to study their own genomes to know is there something different happening in their genomes or not. And that's the way that they like to pair the data and see. They like to see what is happening between the trial, father, mother, and the child. Deceased one, and try to study that out of it. So another technology that we talk about is implicit designer. Okay, implicit design is a place where we have various panels that we can take them up. There's already pre-made panels, so we are ready to use panels that we talk about. So everything that is oncology, disorders, there is a list of panels that have been studied and kept. But with that, we have certain panels which are made by communities. Doctors, scientists who have already worked on certain diseases, disorders. They have made their own panel and provided to the community. We are calling it as community panel. So that is also ready to use. So different. So it may be hearing loss, right? It may be your cardiovascular diseases. We have these panels coming in over there. So they have studied from long time. With that, you can have something called as on demand. So disease-oriented genes are taken up, studied genes. and clustered all together so that people can come, make their own panel out of it for tomorrow's like tomorrow's studies. Like I told, 100 genes, right? So if I know in India, I need to work on thalassemia, need to work on, say, Waters disease, we need to work on, say, SME, right? So, muscular dystrophy. So, you need these genes, if you want, you can cluster them all together, make one single panel out of it. So, on demand. So, somebody would like to use smaller set of genes and try to study their own, make their own study out of it. Why? If you have 100 of patients coming, where you like to screen the genomes out of it, which may be related to SME, which may be related to thalassemia. These information could help you further downstream analysis. Okay. But with that, something called as made to order. This helps you to create your own panels, giving your own genomes. So microbial genomes, okay, or sometimes viral genomes. You can give your genomes, try to design primers for your own genes or genomic regions. So that is called as made to order. I'll show you a demo tomorrow. But this tool is available for iron torrent data, where you can make your own panel out of it. Then there is some powerful tool that is available called as AgriSeq. Okay. Now AgriSeq is something agrigenomics. You like to play with genes or genomic information where you like to have panels which would give you genotyping information. Maybe from the topmost species that you have got to the level of where you have put any genes, inserted any genes, or you want to see the progeny of the lower versions of the gene, plants out of it. I like to look at those areas and then try to pick which is the best one out of those to take it further for production also. So this is like study those genotypes, right? SNP changes, and then try to apply them and get an output for it. And understand which are the best species that we are getting for plants. So it may be anything, sugarcane, it may be even the flowers that we have right now. So genomic, GM flowers are also there, coming right. Yes. So these varieties, how do you like to come to a point where they will be regularly giving me a growth without any problems of those what we say drought or the dryness of the weather, right? It should give me a proper plant as an agricultural. That's where they like to study the genotypes. Then the RNA expressions, right? RNA whole transcriptome is what we started with, but what I could do, I told you we have a technology MPC designer. If you give the gene information, I could design a custom RNA panel for it. So it's for human particular, but you can create a custom panel for those RNAs. You can create for expressions over here. You already have a community panel available who has already studied it. So these are all amplicons which you can use for your own samples to amplification. Now with that, already gene expression kits are available. Somebody who tests somebody for drugs, for metabolism, you like to also test it. Say for 16 years you have text the gut microbiome. For gene expression, you like to look for implicit transcriptome human gene expression kits. So you have two. One is directly human, one is mouse. So mouse model can be also studied over here. Treating the mouse and in your wet lab you are trying to take the gene expressions out. But with these all numbers, you still have the whole big one, whole transcriptome. So whole transcriptome would be total RNA base, where you are trying to look for coding regions, right? You are trying to look for non-coding regions, right? And using that information, the ribodepletion has happened. Ribosomal RNA is removed either, so that you get rest of the information out of it. And use that to map it onto the reference genome, HG90. You have already annotated information. Annotated means you know where exactly onto the genome the information is present. And use that to quantify. Or you use that to know the isoforms. Isoforms is where the transcript should be formed. So if you have 10 exons, the transcript should be formed by 8 exons only or 5 exons only. So you could get to know by doing sequencing which are the transcripts that you are getting after splicing. Okay? So that's the technique of total RNA kit that you have. The other thing that you can do with the whole transcriptome, the gene expression or the isoforms should be for fusions. So a higher version, fusions is in cancers, normally or translocations happening in the chromosomes, you can get to know about fusions that are there. So between the genes, there would be fusions. So you have need more throughput for it, 80 billion reads and so on. With that small RNA. So small RNA would be mRNA that you are targeting, right? So it is more likely that you can follow certain steps that allows you to amplify or pick small RNA from the samples and use those amplified or picked DNA product and go further with sequencing and get the outputs out of it. And the same output you are going with certain steps for doing the analysis. Okay. So small RNA, of course, what would help you? So knock out genes, right? So take that studies further. The gene is working, knock out them, and then try to look further what is happening in the sample. Okay, with that, how the RNA would be more precise is with the total RNA, when you are doing an RNA depletion, that is one, but you can do a selective one, poly A region. So only tagging poly A and segregating them out. So certain studies needs poly A based tagging out. So you must be knowing, right? When your transcript is formed, the end you have poly A regions in here. So there are certain tagging methods where poly A will be picked up and you are sequencing them. You have other methods where that is you are looking at the size modification. Okay, tries to look for miRNA base, or you are looking for missing transcripts, smaller cut transcripts, or you have something like protein binding, RNA protein binding based studies. So use that for epigenetics. Binded protein, dissolving or denaturing entirely DNA, then dissolve the protein regions also. Whatever remains back as double stranded would be again denatured with single stranded and used for sequencing. So that would go for your epigenetic studies. In that way, your RNA. Okay. So with that approach of whole genome, what is the approach of whole genome over there? So you have two things that would come in. One is de novo. You don't know anything and you like to build something out of it. But de novo could be where you have looking at overlapping regions and then building a genome. It would not be a complete genome, but it would be like contigs. We talk about assembled contigs. Sequences that get assembled are called contigs. And once the contigs are assembled together, they are called as scaffolds. Okay. We like to study functions. What is the partial or complete function out of it? So comparing with the databases, NCBI databases, NR databases for proteins, and then trying to pick that function out of it. So you get this by assembly, get the data, take it further with NCBI class. Okay. Or you have reference-based data. References, you already know reference, NTB as an example. You like to align the data because you need to know whether the coverage is sufficient to cover all the regions across the genome. And with that, detect variant out of it. What it would happen, it will give you information on the strains. Or you like to take that and go for typing of those samples. HIV typing, if you have heard this word as HIV typing, you like to differentiate what is the HIV virus out of it. Okay, so there are certain kits which amplify HIV, take those regions and go further with typing of those data. Hyrex is one of the online tools. Or you have Cosmos ID, which helps you to do analysis of the de novo assembled data. Metagenomes. People working on metagenomes. They do what there is lots of step behind it. When you do an assembly, you like to assemble that, see what is happening, how many contigs are formed. Are we able to do a completeness? That means any genome is going to be getting out of that or not. Genome completeness, 80%, 70%. If the genome is there, it is having an initiation region or not. Because where initiation is there, you will find the replication of those bacterial DNA. Okay? Or out of this bacterial DNA, you like to look for bacteriophages. You look for viruses-based particles, right? So you're doing multiple things at the same time. Okay. So, whether आइकन लगाया होगा स्क्रीन सेवर इस करना ना so two types of techniques but what could we do is something like this short reads right and with that short reads okay it can assemble into contigs Again, the same assembly could be paired together by paired and reads, assemble again and make more scaffolds out of it. And the same scaffolds would actually make up a genome if you apply with the backbone of a genome. So you try to look for a closest genome and try to map that and make a genome out. So circularize the genome entirely. So this is a process of doing the work. Okay. But with this assembly, you like to study them more. I spoke about coverage, right? The coverage is whatever you try to look as reads that are overlapping. That tells me what is the overall coverage. Okay. So like number of reads, average read length, and the length of the original genome. So if I say I understand that I have a genome which I'm sequencing maybe around 5 MB. So how much type of coverage I will require to cover that area? So if I need 100x coverage, what is happening 100x? So 5 MB into 100 would be 500 MB of data that is required to do an assembly of that data, right? So when I'm requiring 500 MB, how could my sequencing help me? How much data should be generated? I get that question over. So when I'm thinking as a whole genome, de novo, I like to think, okay, can I generate 2 million reads which have a mean read length of 300? What would be the MB generated? Right? 2 million reads into 300 as the mean length. That gives me 600 MB data. So 600 MB data is sufficient enough for me to take it for a de novo assembly. So 2 million would be sufficient for my system to be generated with a 400 base pair sequence. Okay, that's how we are calculating to know about genome. So this is when you try to think what could you do further. Reference based, as I told you, mapping data, you have a tool called a schema that is inbuilt of the system, okay, which helps you to align the data to the reference. But with that, the online tools that are available, Bouty, BWA, okay, these tools helps you to do alignment through command line. Okay. Then with that alignment, you like to study genome coverage, right? So from our system, we are doing using coverage analysis plugin. Gives you overall idea. But with that, you have plugins on Use Galaxy. Okay. Those plugins helps you to take your own RAM data, try to cover those regions. Which are the regions covered? What is the X coverage mean there? Okay. Average coverage of those regions. You get it over here, Use Galaxy. With that, when you look at variants, you like to call variants, right? And try to see which are the variations that are observed. So the tool on our system is variant caller, which utilizes torrent variant caller program, take all variants. But at the same time, GATK is available, standard one, or you have rest of the tools available on Use Galaxy, where you can use your reference, use a reference, map it, use that map data, and go forward with variant analysis. So that makes up your pipeline for doing the analysis for NGS particularly. Okay. Am I fast? Okay. So this is something like coverage analysis. Tells you about what reads are. Like you can see here, Mycobacterium segmentis strain is there. Okay. I'm trying to look for reads. 4.8 million reads generated. 191x average base coverage. 191. It's not clear, right? That much. But 191x coverage is average coverage. And uniformity, how better across the regions the bases are covered. Uniformly. So it tells me 92%. So how does that happens? It tries to take 20% of this data. So if I'm saying 191, I'll take it the 200x. So I'm taking 20% of that data. And with that 20%, 200 of 20% of 200, how much? Okay. 40, right? So easy. So I know you're drained out, but it's okay. This is what you remember at night, particularly. So with 40, it will try to take as minimum value of 40 and screen all those positions in your genome where you're mapping. And it tells you 92% of the region is above 40x. That's uniformity. Uniformly covered. So एक स्कूल में बच्चे लोग जब जाते हैं तो यूनिफॉर्म पहन के जाते हैं. जो नहीं पहना रहता वो क्लियरली डिस्टिंग्विश हो जाते हैं, राइट? हमने यहाँ पर भी करना, हम पूरे जीनोम में देखना चाहते हो कि अगर हम सीक्वेंसिंग कर रहे हैं इक्वली कवर्ड है कि नहीं, एम्प्लीफाइड इक्वली है कि नहीं, राइट? सो इसके लिए कवरेज एनालिसिस इंपॉर्टेंट है. सो टेल्स यू डिफरेंट ग्राफ्स, क्रोमोसोम वाइज बताते हैं, यूनिफॉर्मिटी बताते हैं, ये ग्राफ बताते हैं कि किस तरीके से प्लॉटिंग हो रहा है ओवरऑल डेटा. एंड विद दिस डेटा यू गेट अ कॉन्फिडेंस कि मैंने जो सीक्वेंस किया है सही है कि नहीं. इट्स मैपिंग, इट्स काउंटेबल एंड कवरिंग दोज रीजंस. ओके, अब ये क्यों कर रहे हैं? सबसे पहली चीज, अगर किसी जीनोम को आप टारगेट करना चाहते हो या सीक्वेंस करना चाहते हो, जैसे फ्रेश सैंपल है माइक्रोबायोलॉजिकल, अब वो लेके सीक्वेंसिंग कर रहे हो, स्टडी एनी कंटेंट, तो आप जानना चाहोगे ना कि जो आप यूज कर रहे हो सही हो रहा है कि नहीं रेफरेंस के साथ. तो आप यहाँ पे रेफरेंस के साथ मैप करके देखिए, and that gives you confidence कि हर सैंपल में डेटा सही आ रहा है. And this would happen for each sample. Okay. So variant calls. So if I'm doing any variant calls, it will take the reference, map it, and give you the results. So this happened in a virus condition. So this is a sample for dengue. Dengue के extraction किए थे and sequencing करके यहाँ पे align किया था. And with that, with the sample controls that they had, they didn't take one sample. With that, it started giving me, so it's completed. Tell me there are 159 variants that are coming in. So SLP, indels, insertions, deletions, insertion extra आ गया, एक delete हो गया. अब ये टूल कहाँ यूटिलाइज किया गया था? SARS-CoV-2. So जब SARS आया था, कोविड आया था, यही टूल्स यूज किए थे. कवरेज एनालिसिस ऑफ वेरिएंट कॉल. वेरिएंट कॉल हो गए, इन्हीं वेरिएंट कॉल से कंसेंसस बनाया गया. कंसेंसस ये होता है कि जहां जहां चेंजेस है ना, तो रेफरेंस ओरिएंटेड जहां जहां चेंजेस दिख रहे हैं वो रिप्लेस करा. वंस रिप्लेस मेक अ फास्टर फाइल. क्या होगा? वही जीनोम है पर जहां पे चेंज है, चेंज रिप्रेजेंट कर रहा है. तो इसी जीनोम को जब कहीं कंपेयर करते थे तब हमें पता चलता था ओमिक्रॉन है, ओमिक्रॉन के वर्जन है, वेरिएंट्स आ रहे, व्हाट इज हैपनिंग फर्दर. ओके, वो भी है आगे. बट दिस इज हाउ यू लाइक टू टेक अ कॉल फॉर योर वेरिएंट्स. ये देखो SARS-CoV वही आ रहा, हमने यही किया था. परफॉर्म प्राइमरी एनालिसिस, राइट? सीक्वेंसिंग कर लिया, डेटा आ गया, रिपोर्ट जनरेट हो गया, बेसिक क्यूसी कर ली थी सिस्टम पे ही. सिस्टम के बाद में सैंपल वाइज क्यूसी, कवरेज एनालिसिस, वेरिएंट कॉलिंग, वेरिएंट के कंसेंसस जनरेट कर ले. सो फास्टर फाइल आ गया, इनकी जीनोम्स आ गए. वही जीनोम आगे जाते हैं, उन आइडेंटिफाई लीनिएज, किस टाइप का वायरस है, किस वर्जन का है, किस लीनिएज का है. तो दैट यू गेट ओवर हियर. एंड क्लैड इंफॉर्मेशन ऑफ फाइलोजेनेटिक्स. ये अलग से सॉफ्टवेयर अवेलेबल है, ऑनलाइन अवेलेबल है. अगर आप GISAID करके एक साइट है, रजिस्ट्रेशन फ्री है, वहां पे उनने सारे इनके जीनोम्स डाले हुए हैं. ओके, सो SARS-CoV के जीनोम्स अवेलेबल है, वहां पे आप देख सकते हो, प्लीज देख सकते हो, विजुअलाइज करा सकते. एक और एक है ग्लोब CoGlu करके, COB-GLU, CoGlu. ओके, वहां पे भी ऐसी स्टडीज की इंफॉर्मेशन डाली गई, आप स्क्रॉल करके ब्राउज करके देख सकते हो. CoGlu वो यूज भी होता है एनालिसिस के लिए. और थर्ड है, देयर इज वन कॉल्ड एज नेक्स्ट्रेन, NEXT नेक्स्ट्रेन करके साइट है. .org है, .com है, पता नहीं है, .com के शायद से नेक्स्ट्रेन करके है. उसी में नेक्स्टक्लैड करके आता है. That's a program to show you how these phylogenetics are there for SARS-CoV-2. और आज भी वो डिटेक्ट हो रहे हैं, आज भी जरा उसके चेंजेस आ रहे हैं, ऐसा नहीं कि वो बंद हुआ. There are things happening. Okay, adventitious viruses. So workflow मतलब papers available है. The paper is simple, like वो collecting sample, extraction हो रहा है, centrifugal force के basis पे और प्लाज्मा सेपरेशन के बेसिस पे वहां से आपका DNA का RNA एक्सट्रैक्ट होता है. वही RNA का कन्वर्जन हो रहा है, लाइब्रेरी बनती, सीक्वेंसिंग दिखता है. एंड विद द बायोइन्फॉर्मेटिक्स पाइपलाइन either using a reference or either taking the RNA and mapping directly to the RNA databases. So जितने वायरसेस के डेटाबेस है 4000 5000 सारे उठा लिए, मैप करवा. सो मैप करने के बाद पता चलता है प्रोफाइलिंग क्या आ रही है. सैंपल में अगर कुछ एक्स्ट्रा बचता है तो उसकी असेंबली की जाती है. सो अ कॉम्प्लेक्स नेचर, जो मैं सिंपल बता रहा था कि कभी जीनोम असेंबली कर दो, कभी रेफरेंस बेस्ड कर दो, अब ये कॉम्प्लेक्सिटी में गया कि अलाइन कर लिया, अलग कर दिया, जो बचा है उठाया असेंबली कर दिया. What would happen? You will get to know information from profiling, you will get to know information by removal. Okay? So इस तरीके से कोई एनालिसिस करता है. ये वही है कंसेंसस वाला फॉलो. सो ये हम फॉलो करने वाले हैं कल 16S के लिए पर्टिकुलर. तो 16S किस तरह से किया जा रहा है कि you have the software getting a report, right? But and report the software is there onto the system itself, which uploads the data to the account. So IAM reporter is the software, actual software. So with this software, once uploaded, it will execute the workflow called as 16S metagenomics workflow 1.1, which would help you to analyze the data, which would give you the results taxonomy counts. So जो आते हैं sample wise taxonomy counts आते हैं, प्रॉपर डिस्ट्रीब्यूशन है विद वेरिएबल रीजंस जो हमने यूज किया वो भी आता है, उसके साथ हमें कंसेंसस क्लब करके भी आता है. ओके, विद दैट ऑल्सो ट्राई टू स्टडी गिव यू ग्राफ क्रोन डायग्राम्स देता है. क्रोन डायग्राम है, डाउनलोडेबल फोल्डर देता है जिसमें बार चार्ट्स आते हैं. ओके, फिर करता है अल्फा बीटा डाइवर्सिटी. सो हैव यू हर्ड दिस वर्ड अल्फा बीटा? अगर सभी को यूनिफॉर्म पहना जाए तो दे विल बी लुकिंग सेम, राइट? I don't want to be very racial kind of thing, but see, think of that, if everybody would look almost same but the faces are different. Now with that, I like to differentiate maybe some certain statistics के उसमें count, जैसे species में counts कैसे आ रहे हैं, कितने वेरिएबल है. सो माइक्रोबियल कंटेंट में ऐसा भी हो सकता है कि दो स्पीशीज डोमिनेटिंग है, बाकी सारे नॉन डोमिनेटिंग या लोअर काउंट्स में आते हैं. इट मे हैपन बिकॉज इट मे बी डिजीज ओरिएंटेड. कुछ ऐसे रहेंगे जो अच्छे गुड स्पीशीज, जैसे 20%, 10% एंड देन 5% एंड 1%. सो ऐसे भी प्रोफाइल रहेंगे जो ज्यादा डाइवर्स होंगे और कम प्रपोर्शन में मिलेंगे, राइट? माइक्रोबियल कंटेंट है वो, और मिलना चाहिए. That's how it should be, because that explains your good microbiomes. So gut microbiome अगर करते हो तो that as a profiling would be very clean. But variability जब आती है, species में बहुत ज्यादा count दिखाई देता है, that means something is extra happening. So giving an example, Pseudomonas कहाँ मिलते हैं? Any idea? Soil में है, right? Soil में है. और कहाँ मिल सकता है? वॉशरूम्स सबसे बड़ा प्रॉब्लमेटिक जगह है. सो एनीथिंग इफ योर लैब, सो दिस इज एन एग्जांपल बिकॉज इफ माय लैब एंड द वॉशरूम इज जस्ट नियर बाय, द टू डोर अवे, और मैं यहाँ पे काम कर रहा हूँ, तो एयर जो है ना, एयर ऐसे घूम के आती है और यहाँ पे ये लैब में मैं काम कर रहा हूँ, बट ना वो एयर की वजह से जो मैं एक्सट्रैक्शन कर रहा हूँ ना, एयर के अंदर का भी डीएनए जा रहा है. अब उनके पास लैमिनार नहीं है, समझो, एग्जांपल आई एम जस्ट गिविंग. सो ही इज नॉट डूइंग ऑन लैमिनार. वो एयर जो आ रहा है ना, वो भी वहां पे एक्सट्रैक्शन में आ रहा है, और जो एक्सट्रैक्ट हो रहा है विद द पीसीआर, ओके, वो वहां से आ रहा है, सीक्वेंसिंग हो रहा है, और रिजल्ट्स पे भी आ रहे हैं. तो वहां पे सबसे ज्यादा भर के सुडोमोनास आ रहे हैं. दैट्स हाउ इट इज हैपनिंग. आई एम जस्ट गिविंग एन एग्जांपल. बट इस तरीके से diversity, where you are trying to see how diverse the sample is within itself. तो अगर हमने देख लिया कि स्पीशीज कैसे आ रहे हैं, काउंट्स कैसे आ रहे हैं, तो उसमें डाइवर्स कितना है एक दूसरे से. मैं अगर बोलूं कि 100 स्पीशीज मिल गए, राइट, चलो सैंपल में 100 स्पीशीज आ गए, बट प्रोपोर्शन में कैसे है? या एक ही जीनस के 10 स्पीशीज आ रहे हैं उसी के अंदर, एंड रेस्ट ऑफ देम आर कमिंग एज सच. सो हम प्रोपोर्शन में देखना चाहेंगे, राइट, कि कौन से जीनस का सबसे ज्यादा आ रहा है. सो ये स्टडी यहाँ पे डाइवर्सिटी, अल्फा डाइवर्सिटी वुड टेल यू whether it's highly diverse sample or highly conserved. So अगर 100 नहीं आ रहे, 10 स्पीशीज आ रहे हैं, so that is going to be very conserved kind of thing. 100 स्पीशीज आ रहे हैं और काउंट्स अलग अलग है, that would be diverse kind of thing, because सब स्पीशीज अपने आप को वहां पे they are trying to perform. That's how it is. So that is alpha. Then there's a beta diversity is diversity between the samples. क्यों करना चाहोगे आप? देखना चाहोगे सैंपल्स कितने सिमिलर है. हाउ सिमिलर? उनमें सिमिलर टाइप के स्पीशीज या जीनस या फैमिलीज ग्रो या डेवलप हो रहे हैं. ओके, ये कहाँ हो सकता है? एग्जाम्पल, कैंसर में अगर कोई सी कैंसर स्टडीज कर रहे हो तो पर्टिकुलरली कैंसर के ग्रुपिंग में आपको ऐसे कुछ दिखाई सकता है. या फिर ड्रग थेरेपीज, पर्टिकुलर टाइप के ड्रग थेरेपीज दे रहे हैं, उनमें 16s करो तो पर्टिकुलर टाइप ऑफ पीपल, when I say particular, it's more like metadata, it's more like differentiation कि किस टाइप के पेशेंट्स है, किस टाइप के यूजर्स हैं. समझो जो खेत में काम कर रहे हैं और जो कंप्यूटर पे काम कर रहे हैं, उनका माइक्रोबायोम अलग होने के चांसेस ज्यादा होते हैं. अगर उनमें कोई डिजीज है, डिसऑर्डर है, तो वो भी और डिफरेंशिएट करेगा. सो ये सब स्टडी है with 16s. This is what we need to explore. Okay, so beta diversity would let you know how the species are coming close to each other or differentiating. With that, how exactly the workflow works, I explained earlier also. The workflow is more like taking your reads, looking for primers. Both the primers है कि नहीं, V2 region, V4 region, V8 region, V6, 7 primers होने चाहिए, तो ही वो आगे पिक होंगे, तो वैलिड रीड्स है. अगर प्राइमर्स है तो वैलिड रीड्स है. यही वैलिड रीड्स आगे जाके क्लस्टर हो रहे हैं, क्लस्टर्स. इफ दे आर गेटिंग क्लस्टर्ड एंड काउंटेड, दोस क्लस्टर्स इंडिविजुअल दैट आर देयर, कॉल इट एज OTU, ऑपरेशनल टेक्सोनॉमिक यूनिट, एंड दैट शुड बी अबाउट 10 रीड्स. वो काउंट है, मिनिमम काउंट. अगर वैसे रहेंगे तो ही वो क्लस्टर आगे यूटिलाइज करेंगे फॉर असाइनिंग अ रेफरेंस, असाइनिंग अ टेक्सोनॉमिक. सो मेगाब्लास्ट करके टूल यूज होता है यहाँ पे. You must have seen Megablast, right? Closest continuous alignment के outputs देता है. There is discontinuous also, and there is a normal Megablast also. So like to do very close alignment of the hit and try to see the topmost hit. With those topmost hit, generate a data report. So it could be HTML, text, and first of it. और यही कल हम देखेंगे 16s में. आउटपुट यही चीजें थी जो मैं एक्सप्लेन कर रहा था. So I need both of the primers. The length should be aligning to the database more than 90% of the length of the transcript. Genus 97% identity से लेंगे, species 99 से. There's something called a slash ID reunion. जब 16s करते हैं तो very close species भी hits आते हैं एक ही cluster को. So it could be like 99.8% similarity, 99.7% similarity. So like to see a difference between them. So topmost और second topmost में से difference 0.2 होना चाहिए. Okay? तो ये वो differentiate करके topmost species को assign करेगा यहाँ पे. Species, genus या families पे. So that's how we like to work with the ion reporter software. ये आपको पहले पढ़ना पड़ेगा, समझना भी पड़ेगा. कल मैं डेमो दे दूंगा, but something that you need to read and understand what happens in 16 years. There are documentation which I will be sharing with sir. And then those could be taken up further. Okay. ऐसे क्रोन डायग्राम्स आते हैं टेलिंग यू अबाउट कहाँ पे स्पीशीज आ रहे हैं. जैसे यहाँ पे क्लेमशेल आ रहा है, वहां पे है स्लैश कॉल्स है. सो स्लैश कॉल्स मींस मल्टीपल स्पीशीज विद सेम सिमिलैरिटी, आइडेंटिटी. 100% आइडेंटिटी से सेम स्पीशीज इट्स मिल सकते हैं. दिस इज समथिंग लाइक योर अल्फा डाइवर्सिटी, जहाँ पे लोअर डाइवर्सिटी इज हैविंग कंजर्वेशन, higher diversity is having higher number of species in it. Okay. Then beta diversity, certain metrics, because आपके counts ऊपर नीचे हो सकते हैं. कुछ species के लिए बहुत ज्यादा counts है, कुछ के लिए कम है. So something like Manhattan matrix is there. You have chi-square test है. Euclidean means you like to see the Euclidean distribution in your data. तो ये study करना पड़ेगा, आपको पढ़ना पड़ेगा. उसके से आपको समझेगा नहीं, because how you like to apply those information to your data. So you need to know about the metrics. Or PC plots है, PCOA. So principal coordinate plots. ये association इसलिए करते हैं कि कैसे species या genus के basis पे वो samples are having correlation. Okay. So जैसे samples, ये वाले samples, ये वाले they come very close to each other. But these samples are more eliminated, or I'll say very far away from them. सो इनके अगर मेटाडेटा मीन्स उसके रिलेटेड जब फीचर्स स्टडी करते हो ना, वो फीचर्स बताते हैं कि वो कैसे अलग है. people having, say, different habits, smoking habits, alcohol habits. People are more sedentary. People are non-sedentary. People are working with computers or non-computers, or people are more physically working. If they are fit, so you're trying to collect this information entirely for your 16S. So I talked about gut. It can be for your soil also. Soil could be different things that you can try to do. Okay. So in this way, you're collecting information, and later on, after you get all this data, you like to compare. Compare between groups. Get those counts of those conditions, compare them. What would happen? It will start telling you which is highly occurring one, which are very low occurring one. So what is the normal has and what does the actual condition has? And that would give you more emphasis on writing your papers also. Okay. So that's how you like to use 16S data and come to this point particularly. So output that is coming tomorrow will be seeing biome format, which can be taken up in downstream analysis. So I spoke about PyCrust, functional analysis. It uses this biome format. So you can utilize that and run it. There's text format which can go forward with the counts. Okay. Those counts could be taken in SAS, SPSS tools, right? Which could help you to do statistical analysis and group the data for your choice. You have other graphs that are coming or results that are provided in family and genus and species. Your FASTA file is provided for you. FASTA file for the OTVs, OTUs actually. So your family, genus and species based. If something is not mapping, there is chance there is new novel species coming, not hitting, or partial reads coming in, right? So in such situation, it will remain as unmapped. So you'll get that away. You also have sequences represented in variable region V2, 4, 8, V3, 6, 7, and 9. So you get that FASTA file. Okay. So with that, the alpha beta diversity matrix and the plots for it would be also in separate folders, zip folders. Okay. So that's how the overall output would come for 16S metagenomics. Okay. I know it's very different, right? But we'll be looking at this data tomorrow particularly. So this is where I end. Okay. So any questions that you have or feel you are trying, we can ask. We can do it tomorrow also. Yeah. So
No, I think also I would like to felicitate him. Actually, we made something for token just for remembrance of this particular workshop. So I made particularly for you, sir. So and
thank you. Thank you very much.
so this is just for remembrance that we participate. So so we give for you that will give you tomorrow. So this is that.